Good evening, students. Welcome to your almost second last class. So today we will deal with bioinformatics part. So I think most of you have joined now. We can start with the class. So in today class we will be dealing with the bioinformatics, some basics about bioinformatics, not in details, but something to know you better, like how things are working in it, and what is the basics behind it, and how it could be helpful in computational biology. So we will deal with some biological concept here, brief history, where this all data came, some new words also we will deal in this topic. So we all know that your biophysics, mathematics, biology, they all come into a one category in this Venn diagram, that is bioinformatics. So mathematics, biology, and biophysics, they're all coming together to make up bioinformatics. So from DNA, we know from the point of view of uh, you know, the dogma, a central dogma of life. From DNA, you make RNA. From RNA, it's a protein. In the DNA, from DNA we can have a DNA genome database. From RNA, we can have complementary DNA transcriptome database. On the protein, we can have protein sequence and structure database. So based on that, we can say about the proteome and phenotype of your sample of your cell culture. So, according to the National Center of Biotechnology (NCBI), bioinformatics is defined as the science of is a field of science in which biology, computer science, and information technology they merge into a single discipline. So, the ultimate goal of the field is to enable the discovery of new biological insights, which unify the principle of biology. So there are three important sub-disciplines within bioinformatics. So first is your development of new algorithms and statistics to assess the relationship among members, and the analysis of interpretation of various types of data, including nucleotide, amino acid sequence, protein domains, and the development and implementations of tools that enable efficient access and management of different types of information. So this is the tree of life. We can see archaea, eukarya, and bacteria showing various subcategories of Crinocotia, Eukaryotia, back, back 1, bowl 3, Euglonosia. So all these are coming into that part. Then there are two types of cells in all we all know that is your eukaryotic and prokaryotic. Prokaryotics includes your bacteria, and eukaryotics are normal human and plant cells. So according to the central dogma of life, you can see gene A and B is producing coding region A and region B. Uh, uh, and from this DNA, your mRNA is being produced, and then from mRNA, a protein is being produced. So both these things are different in prokaryotes and eukaryotes. On the left, you're seeing from the prokaryotic perspective, and the right, it is shown from the eukaryotic perspective. Alternatively, they could be also spliced with the exons which are we are not required. Then we can predict the what should be included, what should not be included in that. So in the protein silencing, splicing, we can have intervening sequencing (IVS). So extene, intene, extene, exon, intron, exon. So you have protein which will produce mature protein. Then you have exon, intron, exon producing RNA. Then mRNA splicing, then a mature RNA out of it. And basically, in this all central dogma of life from DNA to RNA to protein, making a proteome and proteomics, dealing with the various transcriptomes, where is this bioinformatics playing a role? Bioinformatics was playing a role into coding of these of DNA and RNA proteins. So what are the replication is being done, transcription and translation? How many amino acids are being playing role? Phenyl, valine, aspartic acid, glycine, histidine, leucine. All these matters a lot, and how they are working. In that all that case, to interpret the data, bioinformatics is the key role playing in it. So at the end we can say bioinformatics is a data management or database structure, analysis and interpretation of data. We can use web-based or standalone tools for the like Perl, Python, Java, and we can develop new algorithms and statistics in this. So if you are aware of many languages like Python, Java, um, you know, C++, so all could be used to make some codings, make some machine learning. Artificial intelligence nowadays, it's quite in. So you can use them for your experiments, which will analyze your data automatically. You just have to code for one, and the rest it will do it automatically by themselves. So we can say in summary, bioinformatics is actually helping us to uncover biological information hidden in the mass sequence, structures, literature, and other biological data, and it's obtain an insight into the fundamental biology of the cells and organisms. So here you can see genome is making transcriptome, then proteome, then metabolome, and giving rise to phenotype at the end. So how this level of complexity works as compared to the computational tools and infrastructures. So here you can see we have a comparative genomics at the initial point. Then we start to work on the base stage of docking. Like you want to check within one protein where is the catalytic center is available. To check that, we do this molecular docking of your drug and protein samples. Then comes your cell pathway network simulations, then molecular machine classical simulation. At the end, the more complex is to understand the molecular based cell simulations. So in this DNA sequence data, like 4 bases A, G, C, T, coding, they are coding or not, so pass into genes, genetic code, and there are signals and patterns matters in that. So here you can see this is a sequence of one protein, and you are particularly interested from C, G, C, C, A, G, G, C till here. So in that case, your bioinformatics might play a role to check that particularly this sequence might be producing some proteins which are showing one phenotype. And if you interplay with any amino acid here, like if you mutate one, then the function will be not same as it was earlier, and have iteration in your signaling pathway. So basically, in this case, friction enzymes play quite big role, which could recognize specific site in DNA sequences. For example, EcoRI1, which binds to a 6-base pair sequence of GATTC, it cuts from this part. And EcoRI could find this sequence in random DNA sequence anytime. So to do so in a sequence, your bioinformatics will play a role that, okay, you have this DNA sequence, and you will cut it from here and make it into two different segments of your DNA. Then there are purine bases. As we know that adenine, guanine, purines are the largest of two types. Pyrimidines is thymine and cytosine. This is some basics of your DNA and RNA genetic code. Then protein sequence, taxonomic distribution sequence. So here you can see that archibacteria includes around 4%, bacteria has 60%, eukarya has 33%, and viruses have 3% of your protein sequence on the Swiss Prot website or unit Prot KB. So in these websites, as we have discussed with the PyMol, you can recognize various protein structures. So this is a particularly, I think, a dimer of one structure. So the same two structures are being joined together with these bondings here. This is tRNA bonding, and this is different. So this is all being done with the help of, first you analyze the sequence of your protein, then with the computational tools of bioinformatics, you can create a prediction structure like this. So might be the structure might be forming like this, and this is also again. So basically, I'm just telling you why and how your bioinformatics is such an important field, and it's playing in each field everywhere. Important part. If this bioinformatics has not been there, it would be very hard for us things to come to a one point that that what we are looking for, what we are working for, is the thing that we are looking for. So some historical landmarks that in 1953 your structure of DNA was identified first, then in 86 your fluorescence DNA sequence, and then in 2003 your human genome project was completed. From 2003 there was a big break in the field of molecular biology. Since the human genome project has been completed, we are able to understand what is since then, so many technologies, many techniques, molecular biology techniques, biochemistry techniques, your analytical techniques came out to play a role since of the human genome project has been done. So in PubMed, bioinformatics, as it has been done in 2003. So since then, in PubMed, if you search for the bioinformatics, so number of publications related to bioinformatics has extensively increased in this tower, this upward scale. So this is computational biology, computational molecular biology, biomolecular informatics, and biocomputing. Then some historical landmarks after the 53 or 2003 in the 1970 in the case of bioinformatics, Needleman-Wunsch algorithm, Chow-Fasman prediction, Smith-Waterman algorithm, Kite and Doolittle hydropathy plots, Pearson-Lipman FASTA, progressive sequence alignment. Then in 1990, basic local alignment search tools are being dealt with it. Um, Again, here you can see the sequence of your protein. Then there's a increase in the growth of the gene bank from 1971 to 2013. So since the